to be able to be visualized on the camera. Apologies for that.
It will work just as well, I think. You can still share your screen. So.
And meanwhile, I will be working on.
Yeah.
It's okay. Recording started.
Yeah.
Sorry, I almost forgot.
Welcome today at the 6th BeTrust Masterclass, which is called Co-Creating Trust Bacteriophages in Livestock. So please, if you're not a speaker, mute yourself, turn off your camera, and if you have any questions, questions are asked at the end of the session, and you can ask them in the chat. Or if you would like to speak, feel free to do so as well. So here you can see the program of today. So the first speaker is Professor Damien Thiry of the Department of Infectious and Parasitic Diseases of the University of Liège. And then the second speaker is Dr. Jeroen Wouters of Phageguard. And then the BeTrust Consortium will share the results of the consumer and farmer co-creation sessions done by Charlotte Boone of Alice Down the Rabbit Hole and Heleen Imschoot, who is Innovation Manager at Flanders FOOD. But first, I would like to give a brief introduction to the BeTrust project. So my name is Nina Kellner, and I work at Flanders FOOD as well, and I'm coordinator of the BeTrust project. So BeTrust is a project which consists of 7 partners. These partners come from 4 different countries. They consist of 4 clusters, namely Food and Bio Cluster Denmark. We also have Flanders Bio and Flanders Food in Belgium and Food Mazie in Spain. Then it also consists of 2 SMEs, being Alice Down the Rabbit Hole in Belgium and Lama in Italy, and 1 research institute, being VIB. So why did the BeTrust project come to life? When we look at the future of Europe, biotechnology is a very important topic. It is promised to create greener industries, reduce waste, and drive a sustainable future in Europe. But when we look at the potential impact that biotech can have, we can see that at least 50% of this impact hinges on social attitudes. So BeTrust really tries to bring all the different actors together to make biotech implementation possible. So which actors do we bring together? We try to co-create with the actors that are affected by the technology, and we co-design in a later phase. So we try to assess the trust barriers that popped up with the actors that are affected by the technology, and we try to come up with principles and measures to address these trust barriers by talking with actors that have a lot of impact, like policymakers, industry, and so on. So the BeTrust project consists of 4 key pillars, also called critical building blocks. You can see them here. I'll go over it very briefly. So the first pillar is theory of change, for example. So what we try to do in this first pillar is really to try to increase our impact of the project from within. So Lama, the partner responsible for this, really tries to review everything that we do to increase the impact that the project is having. Then the second pillar consists of extended risk-benefit assessments. So we actually selected 6 biotech use cases in the project and We carried out risk-benefit assessments for this, scientific ones, and these risk-benefit assessments were then also discussed in the co-creation trajectories, the fifth pillar, and added with perceived risks and benefits that the stakeholders also identified. And then in the last pillar, we have a forum and an ambassador scheme. So here you can see the 6 use cases. So the first one is on precision fermentation, the second one on biomaterials that are derived through biotechnologies. The third one is on biofertilizers and biostimulants. The fourth one is on the new NGT proposal of the European Commission. Then the fifth one is on cultured meat. And the sixth one is on bacteriophages. So each masterclass in the BeTrust project really tries to address one of our cases and also the results of these cases. So now we are at the 6th masterclass, so I guess we can get started with our first speaker. So I will give the floor to Damien Thiry, the first speaker of today.
Thanks. I will share my screen. Just tell me if it's okay.
So we see your screen. Now we can see the presentation mode as well. Thank you.
Okay, perfect. Do not hesitate to interrupt me if anything is wrong during the presentation. Huh? Okay. So thanks a lot for the introduction. And my duty is to talk about the bacteriophages as alternative to antibiotics in livestock in a research perspective, and we will go through an overview of bacteriophages and phage therapy, and then we will go to how it works in human and veterinary medicine, and then on some research program that we had in livestock. So first of all, I would like to start this presentation with a brief overview on bacteriophages, and you all know this famous guy, I guess. So it's Alexander Fleming, who discovered penicillin in 1928. And when he discovered penicillin, he already knew that resistance to antimicrobial will emerge and already exist actually at this time. So he was already aware that resistance is existing in the nature, and then that bacteria will be resistant to this penicillin. And indeed, this is constant trends all over the antimicrobial era, where you can see here on the top the different antimicrobial antibiotics that were discovered and the date of their discovery. And then on the bottom, you can see the first resistance that appeared in clinic, in practical use of this antimicrobial. And as you can see, for each antimicrobial, some resistance emerge against in the few months or years after their discovery, when they were used in the field. If you check the penicillin, the first resistance that was described in 1940, and you can see that there is. You can see my mouse. Yes, the cursor. You see. Maybe someone can tell me if you can see my cursor.
Yes, we can see it.
You can see. Okay,
Yes.
perfect. So the delay between the discovery and the first resistance that was described is due to the fact that the penicillin has to be purified and tested, etc., and then commercialized and then used in the field. And it was used during the Second World War for the first time. So I like this video. It's a video from another research group who used mega Petri dishes. and they inoculate on these petri dishes some antimicrobials at the extremities. Some E. coli at the extremities, and they divided this big agar in several compartments, as you can see, and they put some antibiotics within this compartment in different concentration. No antibiotics at the extremities, and then 10 times more antibiotics each time. It was trimethoprim. But I will start the video. So this is the huge petri dishes that they used. You can see here the different compartments. Okay. You can see that in the first compartment, the extremities, there is no antibiotics. Then you can see that there is each time 10 times more antibiotics in each compartment, with a maximum in the center. Then they inoculated bacteria at the extremities, as I told you, and they wait for the grew of the bacteria. You can see here the grew of the bacteria. In the first compartment, it was really easy for the bacteria to grow. Then there is a pause before growing in the second compartment, but you can see that in this compartment you have many colonies that appears, and so the bacteria was able to resist to the antibiotics. Then for the third compartment, you can see also a pause, and then the bacteria was also able to grow on this compartment. And it will be the same story till the end, till the centrum, and so the bacteria each time adapt and resist to this huge concentration of antibiotics. So of course this video was fastened, but it shows you the powerful of the bacteria to resist to antimicrobials. They picked up some colonies in each compartment, they sequenced their genomes, and they show the mutation that appears within the bacterial genome. So just to show you the powerful of these bacteria to resist to the antimicrobial. Of course, it's not always this kind of case, but it shows something really amazing. So a bit of history about bacteriophages. Bacteriophages were discovered firstly by Ernest Hanbury-Hankin, actually, who was a UK bacteriologist, and he showed an antibacterial effect of something similar to phage. And this effect was done by an agent which was present in some weavers in India. Then Frederick Twort and Felix Derel deeply analyzed and studied these agents, and they showed that they are viruses, they are ultra-filtrable, they can be isolated, they are really specific. They showed some plaque lysis on the bacterial agar, and Felix Derel performed the first phage therapy experiment in 1919 with an oral administration of phage to treat Salmonella enterica in poultry. So they are considered as the father of phage therapy. Also, André Gracia, who was a Belgian physician, and he was a professor at the University of Liège also, and he discovered bacteriophages against Staph aureus. And Georgi Eliava, who was an important also researcher within phage therapy. He was a student of Felix Derel at Pasteur Institute in Paris, and then he came back to his original country, which was Georgia, and he created in Georgia the Eliava Institute in Tbilisi, and this institute is still present currently, and they treat patients with bacteriophages in this institute. So this is some example of phage cocktail that exist in the past, And then, as I told you, the penicillin was discovered, and the oxidant totally occulted the phage therapy to treat bacterial disease because of the powerful of the antibiotics. And for sure, the antibiotics were and are still really powerful. Also, the Cold War, which make opposition between Occident and Soviet Union, and so all the publications, etc., were written in Russian. So it was not easy for the Occidental researcher to read them, of course. And also the experiments were not really well conducted, and it was not easy to have confidence in this publication. Then the antimicrobial resistance emerging at the end of the Cold War, and also some new research that were well conducted. For example, these two research made by two vets, Smith and Higgins, against E. coli infection were really well conducted and make a renewal of interest for phage therapy in Western countries. So if you check in PubMed, you can put the keyword bacteriophage. You can see a lot of publication since many years, as you can see. And if you compare with the keyword phage therapy, you can see that the publication emerge really in the 2000, it was the start of this exponential curve of publication. Why? This is due to the fact that bacteriophage were highly studied, but for a lot of fundamental aspects in fundamental research, such as, I don't know, DNA heredity, Sanger sequencing, etc., etc. So bacteriophage were really used in fundamental research, but not in phage therapy in Western countries, and it emerged really in when the crisis of antimicrobial resistance was really present in our countries. What is a phage? A phage, there are different kinds of phage. There is the temperate phages, which are phages that are able to integrate their DNA into the host bacterial genome, and once integrated, they are called prophage. So in phage therapy, we are not searching for this temperate or lysogenic phages, because these phages are able to integrate their DNA into the genomes, and by this way to confer to the bacteria new patterns as virulence or resistance patterns. This is an example of research we made in our lab on the STX phage. This phage contained the STX gene, which encode for the Shiga toxin, and the Shiga toxin is really virulent toxin in E. coli. They cause, for example, the famous hamburger disease in human. Then you have the lytic phages, and these phages are searched in phage therapy because they infect the bacteria and they lyse the bacteria. They replicate into the bacteria thanks to the cellular machinery, and they lyse the bacteria, so they will not integrate their DNA into the bacterial genome. So these phages are searched for in phage therapy. So phages are really abundant. They are the more abundant microorganism worldwide. They are involved in bacterial evolution, and they allow also regulation of the bacterial population into the ecosystems thanks to lytic and temperate bacteriophages. If we have a quick view on the cycle, you can see that the phage will attach to the bacteria, inject its DNA, the DNA will be circularized, and it will be replicated thanks to the bacterial cellular machinery. You have the phage assembly, and then the lysis of the bacteria and the release of many phage into the environment. This is the lytic phage, but some phages can have a lysogenic cycle, as I told you. So the phage attach, inject its DNA, and then the DNA will be integrated into the bacterial genomes, and it will be called prophage at this time. And this prophage will be replicated during the bacterial division to the daughter cells. Of course, this prophage can be also excised from the bacterial genomes due to different environmental conditions. When the bacteria is not in well-being condition, the phage can be excised and enter a lytic cycle to go out of the bacteria and start a new cycle. But in phage therapy, we search for lytic cycle. Also, we will not go into detail in this slide, but just to show you that phage and bacteria can co-evolve. And of course, the bacteria will try to resist to the phage infection, for sure, with a lot of different systems, such as a CRISPR-Cas system, as you maybe already heard about it. This is a kind of immune adaptive system of the bacteria. So the bacteria can develop strategy to resist, but the phage can also develop a strategy to counter the resistance of the bacteria by modifying the receptor binding proteins, etc., etc. So this is really a kind of Darwinian process. We can use phage themselves, but we can also use bacteriophage derivatives. And of course, phage will use enzyme to in different step of the infectious process, and they will use some depolymerase, for example, or some endolysin, which are able to degrade the peptidoglycan. So we can also imagine to use some specific enzyme derivated from the phages as what we can call antibiotics, enzyme biotic antibiotics. What about the phage therapy in human medicine? As I told you, the Eliava Institute was created in 1923 by Giorgio Eliava, and so this institute is still present now in Tbilisi, and they treat patients since decades with phage. Then in Europe, the first phage therapy unit was created in Poland, and this in 2005. And this first phage therapy unit was able to treat patients with phage thanks to the declaration of Helsinki, and a specific article in this declaration, the Article 37, which specifies that a human doctor can use some experimental treatment in last resort treatment, of course. So you have a lot of administrative stuff to do, but they can use phage in this context in human medicine. And this is within this context that the human doctor used phages in Europe since many years now. And but it changed a few years ago, as I will explain you. And you have several examples in the media of phage therapy success. There are not always success with phage therapy, for sure, but you have some success. And for example, here it was a young children who was liver transplanted, and he suffered from an infection. And the human doctor has to treat this really young children with phage therapy during 85 days between to wait for a second liver transplantation. And it was a success, and this young children survived and received his second liver transplantation. So it was a success that maybe you already heard about. But the main break in Europe is the regulatory for phage. As I told you, they used phage as a last resort treatment in the context of the Helsinki Declaration. And of course, if you think to put into the market antibiotics, it will take you more than 10 years because you have to do some preclinical, some clinical, some GMP process, and also you have to receive regulatory approval. So it takes a lot of time and it costs a lot of money. So if we have to do it for phages, it's really not possible because phages are really specific from a type of bacteria. If you think, for example, to Klebsiella pneumoniae, you have more than 70 capsular type of Klebsiella pneumoniae, and some phages are specific to one capsular type. So it shows you that it's really specific, and you have to imagine some cocktail or to adapt your treatment based on the bacteria you have. So it's not possible to do this pipeline for each phages for sure. So in Belgium, in the Queen Street Military Hospital, they worked on phage therapy since decades now, and they wrote a monograph, a general monograph, which was accepted by the government, and which explain how to produce a phage as a phage active pharmaceutical ingredients. It means that this phage will be used as a magistral preparation in a hospital. So this general monograph was accepted by the Belgian government, and the Queen Street Military Hospital since 2019 can produce phages for human medicine, only used in hospital, because this phage has to be handled in the hospital pharmacy, but they can use them without the declaration of Helsinki, so without the context of the last resort treatment. Their challenge were also to include the phage in the pharmacopoeia, in the European pharmacopoeia, and a new general chapter was published in 2024 in the EDQM website, on the EDQM website. So it moved a lot in human. What about in veterinary medicine? Each time I discuss with colleagues, they said, okay, in veterinary medicine, it's more easy to use phages in animals. You have less rules, regulatory is more easy, etc. And I always have to explain that it's really not more easy in veterinary medicine than human medicine, because the animal that we will treat will maybe be on your plate tomorrow. So we have to, we cannot use experimental treatment like that in veterinary medicine, because maybe we will eat this animal. So the regulations are more strict finally in veterinary medicine than in human medicine. So but the EMA decided to include phages as novel therapies. So they published their new regulation in 2019, and they included the phages into the chapter novel therapies. And an expert group wrote the guidelines for these bacteriophages, and they published these guidelines in 2023. These guidelines explain to the companies how to register phage or phage cocktail at the European level, so at the European Medical Agency, to get a centralized registration of this phage preparation. So this is really nice that the EMA decided to include phages in veterinary medicine within this novel therapy, so to have regulation for these phages. But the big inconvenience is that phages are really specific, as I told you, and this kind of centralized registration takes really a long while a long time to be registered, and also it costs a lot to the company. So I'm not sure that this is the best way to use use phage in the field. Maybe the magistral preparation could be the best option, but in veterinary medicine, it's really difficult to move this kind of regulation. So if you check phage therapy, I show you this picture a few slides ago. If you put veterinary phage therapy, you can see that we have really scarce publication on the topic. You can see, for example, in 2014, 259 publications in phage therapy globally, and in veterinary phage therapy only 22 publications. Of course, we have scarce literature compared to human, but we can make a lot of comparison between what is done in human medicine and what is done also in veterinary medicine. And we have also a lot of encouraging literature on the topic in veterinary medicine. For me, the important criteria to take into account when we think about phage therapy is first of all, isolation of the phage, then genomic and biochemical characterization of these phages to be sure that they are not, for example, lysogenic phage, but well lytic phage. The spectrum of the phage has to be studied to see what kind of bacteria these phage are able to lyse. The safety of the phage, the safety for the animal, the efficacy, of course, of the phage, the resistance that could appear against these bacteriophages, their impact into the microbiota, their impact into on the environment can be also studied, the way of administration, the routes of administration, maybe combination with antibiotics, because in human medicine, they always use phage combined to antibiotics. That's why also I don't like the terms alternative to antimicrobials, but maybe more complement adjunct to antibiotics. And then we have to think about the production, the control, and the stability of these phages before they're used. In my lab, we work on this different green circle, and I will show you two research that we made in my lab in livestock. The first one is about phage in post-weaning diarrhea in pigs, which is an important disease in pig production. And in this work done by Margot, a student in the lab, we isolated phages in wastewater. This is a really good source of phages. And we isolated phages against the ETEC F18 bacteria. This is a non-terotoxigenic E. coli which caused this post-weaning diarrhea. We isolated 4 phages. You can see here their morphology in electronic microscopy. We will not go into details in all the data, but as you can see here the process from the isolation to the characterization. We then sequence their genomes, and the first thing that we would want to see when we sequence the genomes of the phage is that the absence of lysogenic related genes. So it's really important, because if we have lysogenic related genes, we can discard this phage for the moment for phage therapy, as I explained. So we compare also this phage to the data bank, and we we can classify this phage into their family, viral family. Then we made a small host spectrum. We tested 2 kinds of ETEC. As you can see here, some 2 kinds of Fimbriae type of this ETEC, and we tried several bacteria. And here you can see the phage. Okay. You can see the phage is able to lyse the bacteria if you have a number. Okay, if you have n, it means that there is no lysis. We decided to continue with this first phage and this bacteria. Okay, for to continue our research, because these phage were really specific, and we wanted to see the effect of a really specific phage. We then try the temperature resistance of the 4 phage, and also their pH, the stability of the phage at different pH. You can see that between 25 and 60 degrees, the phage is stable, and at pH between 4 and 10, the phages are stable also. Then we move to a preliminary in vivo model to see finally how the phage works in an in vivo model. So this Galleria larvae model is Galleria melonella, is a butterfly, a night butterfly, and we use the larvae to see how the phage efficacy and safety within this really preliminary model. So we made different groups, PBS, You can see here the bacteria, so the bacteria alone. PBS is a buffer, so it's just a negative control. Here, this is the infectivity control. Then the phage safety also control, just PBS and the phage. And then the treatment, the bacteria with the phage at different MOI. MOI is multiplicity of infection. So finally, this is the concentration of phage that we will use. So we check the survival of the larvae at different time point during 4 days. So what we see really briefly is that in the infectivity group, you can see that all larvae were dead at 24 hours post-inoculation. And in all the other groups, the phages were able to treat to infect the bacteria, and the larvae were able to survive in this model. Here you can see the black larvae are the dead one. It is due to the melanization. This is a part of the immune system of the larvae, and when they are alive, they are creamy colored. Then we wanted to check the impact of the phages within the intestinal microbiota of piglets. So we took 37 feces material of 37 different piglets at the age of weaning. We mixed these different feces, and we put these feces in fermenters with and we had also control, of course. And what we wanted to check is the impact within the microbiota and also the impact in the on the short chain fatty acid. So we really briefly showed no significant effect of the phages within the short chain fatty acid and within the on the intestinal microbiota by metagenetic analysis. Then a second study that I would like to talk about, and I will do it quickly because I'm already at the end of my presentation. It's about phage therapy in the context of bovine mastitis caused by Staph aureus. It was done by Cyril Ngassam, who was a former PhD student in the lab. And here we used a phage that we inoculated to murine mastitis model. Here you can see the first group. This is the bacteria and the phage. Then the bacteria and an antibiotics, marbofloxacin. Then the bacteria alone as infectivity control. Then the phage safety control, and then the negative control group. We inoculated the bacteria and the phage 1 hour later, the bacterial inoculation, and we euthanized the mice at 1 day and 2 days post-inoculation. What we observed, if you check here the negative control, you can see here the hour post-inoculation 24 and 48 hour post-inoculation. You can see the 4 mouse, and you can see the bacterial and the phage titer. So in the negative control, of course, we didn't find any bacteria, neither phage. So this is nice. In the ISP control, so the phage safety control, we of course didn't find any bacteria, and we found some phages that were able to be present till 48 hours post-inoculation. Then in the infectivity control group, we can see a lot of bacteria, and the bacterial titer increased between the day 1 and day 2 post-inoculation, and we didn't find, of course, any phages. In the antibiotic treatment, we can see that this antibiotic was really efficient, but we knew it. We chose this antibiotic because it was efficient. So we can see that the bacteria was still present at post-inoculation 24 in 1 mice, but no bacteria found at 48 hours post-inoculation, and no phage for sure. And if we come to the phage treatment group, we can see that we observed bacteria in the mouse at the first day post-inoculation, and we in 3 mouse, in 3 mammary glands and 2 mouse, we can see that there is no bacteria anymore. We just found a little bit of bacteria in one mammary gland of the fourth mouse, and but the titer was lower than at first day post inoculation, and that then at the in the infectivity group. We also observed that in the 4 mammary gland studied, we observed phages at our post inoculation 24 in 3 of them, and no phages anymore at our post inoculation 48, which means that maybe the phage were destroyed by the immunological system. Here you have the picture of the necropsy, and you can see a huge inflammation in the infectivity group compared to the antibiotic treatment group and to the phage treatment group. You can see here more inflammation than the antibiotic treatment group, but less inflammation than in the infectivity group. So this is my last slide. Bacteriophages are the useful weapons against antibiotic-resistant bacteria. There is a lot of advantage, of course, and disadvantage. I explained you that there is really narrow a spectrum of the phage, so this is nice to avoid any impact on the microbiota, or to have a low impact on the microbiota. But of course, we have to imagine to develop some cocktail of phage, because we cannot have a probabilistic approach such as with antibiotics. A high diversity of phage exists. I told you that we can find phage in different environments, and there is a lot of phage in the environment. The mechanism of the phages are quickly bactericide. You can also have antibiofilm activity, and it not depends to the antimicrobial resistance for sure. But of course, you can have also some inconvenience with maybe endotoxin release, but also we have to study the pharmacodynamics and kinetics, which are often unknown. There are virus or bacteria. They cannot infect eukaryotic cells, and they are able to replicate at the site of infection. So this is nice, but of course you can have an immunity, a cellular and humoral immunity, that will be mounted against these phages, and we have to pay attention to their stability, of course. And then for sure, resistance can also appear against bacteriophages. So this also must be well studied. So I thank you for your attention, and I'm of course open for the question at the end of the session. Thanks a lot.
Thank you so much, Damien. Very interesting presentation. We're a bit behind schedule, so let's move on to Jeroen Wouters, the next speaker, and we'll have some time for questions at the end. But feel free to use the chat already and pop your questions in there. Then the speakers can think about them, and at the end of the session, we will address them. So welcome, Jeroen. You're still muted, Jeroen.
That's correct.
Yes.
Unmute here. And do you hear me? I have to apologize for my voice.
Yes.
And I have to apologize that I'm not visible. My camera is not working this morning. So a bit of a puzzle, but you see a Phageguard logo somewhere, right? Am I sharing my screen now?
Yes, we can share. We can see your screen, and we can see the Phageguard at your camera. But unfortunately, we don't see you. But good luck with the presentation.
Yeah, thank you. And we tried everything to get that fixed. I'm now waving everybody and saying hello, but you don't see that. That's a pity. But please bear with me, and happy to share you some insights on the bacteriophage use from the perspective of Phageguard. And as you see, the name of the masterclass was a bit oriented to livestock. I have to give right away a sort of a disclaimer that we are fully oriented towards food safety, so on product use, but I will explain it in a moment. It was a great pleasure to listen to Damien, who highly already indicated the power of bacteriophages and the wide type of uses also of these nice microorganisms. So I'm happy to dive into that. Briefly here, a bit of background, and yeah, I have a food technology background at Wageningen University and a PhD. Worked at NISO, and then also worked on early time phages of What is it, bacteria? I see that the sound is not good. Is that correct?
I noticed that you're sometimes like the connection is not very well. For me, most of the time I hear you, but in the beginning it was a bit abrupt. So I don't know, is your connection good?
Then it should. No, yeah. At my side, it all looks good, but yeah, I cannot do about it right now. But,
Okay, let's continue and hope that it's
apologies for that.
yeah.
Someone knows already, but and working at NISO and doing early stage work and bacteriophages even before Phageguard was there as a company. Then worked on innovation management and also collaborated quite a bit with the colleagues here of Flanders FOOD. So good to see that joint history. And then I joined Phageguard in April 2023. That's about 2 years ago, and I'm involved in the research and innovation activities at our company. Now, and then I will introduce a bit about Phageguard, then dive into bacteriophages, and especially in Listeria and Salmonella control, and some conclusions. And here you see the team. We're with 40 people, a scale-up company we refer to ourselves, and started quite some years ago already. But first, food safety. And food safety, you think, hey, we are much better in controlling all the aspects, but on the other hand, we still see that food safety problems occur and pop up. We are very much US focused as a company, and thus here I highlight some of the recent activities in the US where Listeria was observed on liverwurst, with a lot of hospitalizations and even people dying, but also E. coli is being observed. This was a case of a large chain, and Salmonella is now popping up in a lot of categories. So not only in poultry-oriented settings, but also in fruit and vegetables, and here, for example, in cucumbers. And also for us important is that regulatory frameworks often determine quite a bit on, well, the control and the attention for the specific pathogens. And in the US, a large Salmonella framework is in place. Depends on how this will, it's basically on hold now, and it will mean that it will be going to the previous program. And then also in the EU, if we look at that, we also keep an eye on that, of course. But if you look to the number of salmonellosis cases in the EU or Listeria, you see that it's over the years, this is reported by EFSA, pretty stable, or at least not decreasing. And there are some trend lines here that you maybe even see still an increase in the number of cases that are out there. So yes, we are maybe better able in food production to control items, but it's not going away, and we stay alert on those type of developments. Now, then, of course, we are diving into the breakthrough technology of bacteriophages, and that is also highlighted at other locations. For example, here in the emerging technologies overview by the World Economic Forum, indicating that the potence is there. This was 2024, by the way, 2023. And we are already, like Damien indicated, working on bacteriophages for many, many years, but still it's seen as an emergent technology. And I like to use the term precision food safety a lot, because of the targeted nature of bacteriophages, you can really pinpoint the specific target organism and thus employ bacteriophages in that. Now, this also is a very good one to highlight. There's a lot of other companies also producing bacteriophages, and that is nice because they all have different areas of expertise and different application areas. It also means that the industry is developing and that we are much better able to produce bacteriophages and being able to market them as well. And you see here various names, but I would really like to highlight Phage EU also as a cluster activity or representing the, let's say, phage community. And there's even maybe a bit of an advice to be trust and Phage EU to connect, because it's really crucial to think about those networks for evolving a new technology. So that is highly appreciated in that respect also. Now, then a little bit about Phageguard. We started under the name of Microbials, then went on with the Microbials food safety name. And in 2023, we fully worked with the name Phageguard. Phageguard is also the name of our product. You see here the product against Salmonella, Listeria, and E. coli. And you can see that it is in bottles. So these are liquids and liquids can be applied on food surfaces. We also have other parts which dive a bit more into other activities, for example, skincare or pharmaceutical applications. And that was also highlighted by Damien a little bit. In those cases, also parts of phages can be used, for example, endolysins in this case. But as Phageguard, we are a full separate unit within the Macrios group and really focused on food safety. And that is highlighted here, Dutch company based in Wageningen, the Netherlands, and one of the global leaders. We are able to produce bacteriophages in a very efficient and controlled way. And we are very active also in the US and produce from the Dutch basis. And we have 3 specific products, PGE, L, and S, and I will focus a bit on that. And our mission is really to protect people from foodborne diseases. And the vision is that we use phage technology to do so. And that this is a very powerful tool in the combination of things in hurdle technology to address the topic of elimination of food pathogens. And here you see the products once more, and you can see that in different product categories you will have opportunities to work. So we refer to product market combinations, and then for the specific categories you might end up going to specific products. So for dairy, there's a wish to work more towards listeria, and in poultry there's a lot of salmonella attention. And we have one pre-harvest application basically, and that is the use of E. coli phages on hides of animals. So that is live animals, but that's just prior to slaughter. And also the raw pet food sector is a very important one, and fruit and vegetables, as I said earlier, also there a lot of observation of food pathogens is being made. Now then a bit about bacteriophages, but I will be quick in that part. Abundant organisms living very close to their bacteria and co-evolve. And the fun thing is that they're very targeted and specific and don't harm anything else but that specific bacterium. And that is the power. On the other hand, it is also the limitation. So it's good to be aware of that there's the specificity, but you don't attack any other food spoilers or food pathogen organisms. So it would not disclose you from any other food safety measures, etc. Important to know. Now here the life cycle is indicated again, and you can make very fancy representations of phages, and this was already indicated to Damien, and also highlighting that for the commercial use the strictly lytic phages are being used and not the lysogenic ones. So that is highlighted here. And another nice aspect is that you can quantify bacteriophages in their in the concentration, and it is very important because there's a dose dependency in their mode of action to control that, and we are very active in that. Then it is nice to pick up the example of listeria control, and I will highlight on that. Now here you see a specific phage which is called Listix P100, and that's now the Phageguard L product, and that has a very broad range within the listeria species, and especially on the monocytogenous ones. So that is the product actually, and then you can dive into the contents of the cell wall recognition and the recognition of cells, and we learn a lot about the efficacy and the host range of our phages based on cell wall recognition. And what is also important, Damien also mentioned the term of efficacy. When are phages efficacious? It depends on the host range. How many of the target group do they actually target, but also on the dosage. So how many phages are there, and how well are these phages distributed over the surface or over the target area. And when you increase the dose, there's of course a higher chance of meeting the target organism and also being efficacious. For now, and there's also a price optimum in that, because phages are not so cheap, honestly. And then you can see where your balance of control would be. And then there are other aspects on phages. It's a topical treatment. They are referred to as processing aids, and they are not tasting, so they don't have an organic influence or organoleptic influence. They are also organic, and we have OMRI control for that. And what is nice in the area of comparison to chemical treatments, for example, there's no wastewater issues, no worker safety, and it's very mild to equipment. So they're also in the comparison to cleaning agents, really, different positions in a biological natural tool. Now, and then I said we can utilize the phages on multiple occasions. You can do it on food with the distribution, but you can also do specific treatments of equipment and then treat at the same at locations. Now, but we also, in view of resistance development, we are aiming majority of applications at the end of the pipeline of the food production, so that the treatment of phages is really at the end of the process. By that, we cannot have a repeated exposure to bacteriophages and the target organisms, but then it's, say, end of pipeline and leaving the production environment. And in the production equipment cleaning, standardized protocols are available to rinse off all phages, so there's also not a repeated exposure foreseen. And that is important to keep in mind. Now, that is efficacy is been shown in multiple publications over the years, and there's a whole set of data on various applications of our products, but also others on that. It is very nicely captured and also summarized and reviewed in a relatively recent review, and overall a body of evidence of more than 50 scientific publications on Phageguard L alone. This is also nice to show efficacy assays, and here you see how that is performed in a laboratory setting, but very close to a practical setting. So here we use the example of fish, raw salmon, and make preparations of square cubes, and then contaminate it, artificially contaminated with listeria, and have a phage treatment at a certain doses and at a certain time, and then the bacteria are retrieved, and you can see a control versus a phage treated sample and make comparisons. And then you can see that we easily maintain 90% reductions. Reductions can be higher depending on the dose and the time, but we also would like to be fair and then say, hey, there's a 10-fold reduction is very well applicable. And what is also nice is that you can look in various sequence type of Listeria monocytogenes, and then also you can see that the efficacy is noted for the whole set of sequence types that have a relevant in food industry. So that is an example of demonstrating that, but also assisting companies in that. Now, and then a very important topic on phage application is approvals, or the regulatory framework. And we see that a lot of countries have adopted approvals for our products, PGS, PGL, PGE, and especially in the US, it's widely used with FDA cross approvals and USDA approved use in the specific product categories. Canada and Australia and New Zealand follow that, and more companies are popping up, India and Egypt. And here in PGL, you see also similar, and we are very active working on the approval of Phageguard L in the EU framework, and I will highlight that a little bit more. And for PGE, it's a smaller product for us. It is a bit more focused on that height application. And this is, by the way, an 0157 specific serotype oriented product. Now, and then for EU pathway, we are very active on the dossier regarding Phageguard L, and there is a long history behind this, honestly. But in 2016, a very nice EFSA opinion has been written and published on this for the use of PGL on ready-to-eat food products of animal origin, and that basically assesses the safety, the need to have such a solution, and also the efficacy. And that is a standpoint. At that time, it was a bit unclear in what category our product should fall. And now it has become clear pretty recently that we should be registered as a decontaminant under a specific regulation, which is 853/2004. And for that, a draft regulation has been written. And currently we are in the process of updating the 2016 EFSA review. It's close to 10 years of new information. So that is currently ongoing. And then we expect to have the updated EFSA opinion in 2005. And to update that EFSA opinion, that is very helpful for the DJ Santé, who is guiding together with us the regulatory decision process towards hopeful anticipated approval in 2026. So that is a bit of a lengthy pathway, but now it's clear in which category we will be falling in a decontaminant, at least for the situation of a ready-to-eat animal origin product. Things are clear. And then you can understand that in other categories or for other uses, again, regulation will have to be followed. And you can see also that it is a very specific phage-oriented pathway. So it is tedious and controlled. On one hand, you see maybe a bit more liberal position in the USA. On the other hand, it is also good to review the different perspectives from an EU standpoint. So that's where we're in the midst of. Now, looking at the time, I will briefly also mention Salmonella. And Salmonella is, of course, a big thing in the poultry sector, but also others, as I said. And for that, we also have a specific product which is named Phageguard S, which is a cocktail of different phages. And also, well, the serotypes in Salmonella are very wide, and we see the similar type of benefits here for this product. And also here we can easily do phage efficacy experiments, and here the same protocol is highlighted, but then for chicken services, and we can do poultry and others, etc. And there also we see, in this case for Salmonella enteritidis, but also different serotypes, a very good efficacy going up to log 1, 2, 3 type of level of reduction. Now, this was also highlighted in the new framework that would be there. So, diving into that. What is very nice that there also have a lot of real life examples in the US. That's why I also would like to bring out this example. In the US, there's a classification system for poultry processors, which make them go in a category 1 to 3. And here we see the historic USDA reported observations of Salmonella positives and without phage application, and after phage application, in this case for crown turkey in a blender, you see that the number of positives is really going down, and which means that this company is going from a class 3 to a class 1 more better control, Salmonella control company, which has benefits in speed of operation, but also the suppliers that can be supplied. So that is a very practical example, and I really love to show also those examples with you. Now, with that, I come to some conclusions, and I cannot tell you that we are, or should really tell you that we're in the midst of a lot of development and research collaborations with academia, with institutes, educational organizations, and networks, and really thrive on the involvement on that, which builds the momentum for our activities. We are based in Wageningen, so team up also quite a bit with Wageningen University on a lot of educational and, well, development trajectories. And to sum up, we are thriving on that. Bacteriophages are a very nice additional tool in controlling food pathogens, and product excellence is key. So effective bacteriophage product for this control. We co-develop with our customers, the end users, and we know that we have to really understand the phage application in the industrial setting. We're constantly looking to new phages in the discovery pipeline to also target new product market combinations. And for us, collaboration is key in that. So happy to participate in this activity also. So thank you for that and your attention there. Really, as in a Belgium setting, I think I can make this lingual joke, like phage development is a feestje, but we should now stop and quit with all the fun stuff about phages. Thank you for your attention.
Thank you so much, Jeroen. In the end, it all turned out great.
Thank
So
you.
we were able to hear you. So that's a relief. So let me introduce to you the next speakers. They are both partners in the Bee Trust project. I welcome Charlotte Boone from Alice Down the Rabbit Hole and Heleen Imschoot from Flanders FOOD.
So, hi everyone. I hope you can see my presentation well.
Yes.
So maybe a bit about Alice Down Rabbit Hole first. So we are a company that specializes in consumer involvement, co-creation and participatory processes. And in the Bee Trust project, we were responsible for the co-creation methodology and also the setup of the consumer co-creation sessions. So we investigated the perspective of the general public about these bacteriophages, and then our cluster partner, Flanders FOOD, did similar session about the bacteriophages with the farmers to get their perspective as well. So why did we ask the opinion of consumers? Because it is a very strange case, bacteriophages as an alternative to antibiotics in meat production. But we did that because in the end, it concerns the food they eat. And applying this technology on a large scale may affect their environment, their health, and even influence social structures. So they might hear things on media or on social media, and sometimes they may be true or not, but all these things, they will form their opinions. And in the end, they will be the ones who have to buy it, both literally and figuratively, because they have to buy the food. We also involved the farmers, which is probably in this case more obvious why we did that, because it concerns their livelihoods. And they need to keep diseases in their herd or the flock at bay, and they want to do this as effective as possible with the least side effects. So their acceptance is also vital for the successful adoption and practical application. And on top of that, the farmers tend to feel that political decisions are made without their input, for example, when restricting the use of certain products to eradicate pests, and that their voices are not being heard or considered in these decisions. Now, why did we choose co-creation sessions and not, for example, the surveys? First of all, there is already quite some data available from previous projects or that is already being generated in parallel projects. And we want to build on those and dig deeper in the causes of the trust and acceptance issues. Things like what forms their opinions or how do they perceive these technologies, certain aspects thereof, what are their main concerns and how they can be addressed. So we brainstormed with these consumers and we asked them about doom scenarios they have in their heads, but also how they can be avoided. So co-creation allows us to not only dig deeper into these trust issues, but also find solutions for them. So here I will show you the conclusions from these co-creation sessions, which is qualitative research. So bear this in mind while interpreting the results. And we will also still need to validate some of this information quantitatively. And we will do this via specific new surveys. So let's start with the co-creation with sessions with the consumers. And first I will frame a little bit about how we set this up. So we took this case together with the case of biostimulants. Because they both cases were rather difficult concepts firstly, but also they are not directly related to their everyday activities because there is no direct alteration of food products. However, it could still affect the quality of their foods in terms of residues, for example, and have effects on their health and environment. So we gathered a group of 15 people in this case from different backgrounds. And we aimed to have mostly people without a scientific background. This was in all the sessions for all the cases. However, we noticed that for this session, the people tended to be a little more educated on food and food production than for the other sessions. And this was probably because this is a topic that requires a more than usual interest in food and food production to be willing to participate in it and care about the subject also when buying food products. So it is something we noticed for all the sessions because we work with volunteers. We only get participants who are at least a little bit interested in food and food production. And this is fine because these are actually also the ones that may have trust issues when they are buying foods and about a certain ingredients or technologies used like biotechnologies. So we are getting the right target group. What we also did was when we sent out our invitations that we chose our wordings very well. We chose them very carefully. We thought out very well. So we wanted to avoid people who are very strong advocates, like either for or against a technology like biotechnologies. So we did not mention biotechnology at all in the invitation. We made it instead an invitation for an open dialogue about the future of food production, and this without priming or steering their opinions. What we also did was create a relaxed environment and atmosphere by providing them with a full dinner. And this is also in order to recreate a feeling of just having conversations at a restaurant with friends or peers. So in this restaurant environment, they feel comfortable enough to state their opinions without judgment. We also did some other things that they may feel comfortable about stating their opinions. So we made sure there is a mix of individual exercises in small groups, usually with people with similar opinions, so they never felt alone in their opinion, and then plenary discussions. The session's desired outcome was to have the answers to 5 central questions. So this was about setup. So we wanted to know about the general perception, the impacts and the consequences they accept from implementation, and how else the problem could be solved, about how the risks could be mitigated, and what messages and narratives resonate most with them. But we did not ask these questions directly. So we disguised them in all kinds of exercises like association exercises, yes or no group statements, scenario exercises, and open discussions, and other exercises as well. We also give them a lot of information during the session, which is also necessary. And we do this stepwise because we want to capture any changes in perceptions before and after some information is given. We also do a clear framing of the why, so the problems that it can help tackle this, and in this case, it's obviously an alternative for antibiotics to prevent the overuse of antibiotics and develop resistance against them. And then we also explained the technology in layman's terms. The bacteriophages we framed as the natural enemy of bacteria. We also mentioned it was a virus, and we also showed a little video of how these bacteriophages make the bacteria burst. But we addressed this natural enemy of bacteria first, so it was clear to them that it was very specific to bacteria and it could not harm humans or plants or animals. We also tried to be as neutral as possible, so we asked them whether they saw this as a potential solutions rather than stating like this is the new and better alternative than for antibiotics. And we also presented like both the potential benefits as well as the risks. So this was all quite heavy and new to most of the participants. It's a lot of information, but we wanted to give them enough baggage to make like an informed opinion and make their own opinion. So enough about the setup now, and let's get to their responses and what our conclusions from these are. So first of all, bacteriophages are generally not well-known organisms, so they are a bit of a mystery, and it's safe to assume that most people have never heard of them. However, in this we were surprised to see that a few participants had heard of them, but more in a medical context. But none of the participants actually knew what they were, how they worked, or anything like that. So they came across as a bit exotic. They are exciting, especially because they look so cool, but also a bit scary because we did mention they were viruses and viruses can mutate. So obviously they asked the question, what happens when they mutate or attack good bacteria? Now, even though that is unlikely to happen, and probably throughout the millennia that these bacteriophages have existed, it has happened over time. So even if it's unlikely to happen, if you apply them on a mass scale, it will be more likely to happen. So we don't know basically what will happen if you apply them on a mass scale into the environment. What was also clear was that this technology did not face the same stigma as the technologies that were linked with more industrial or laboratory environments, like genetic modifications or cultured meat or cellular agriculture, and that this was rather perceived as rather natural. So as we took this together with the case of biostimulants, for biostimulants, this was even more obvious, but they acknowledged also the phages are something that are just present in nature and they are a natural concept. So it could be that the more agricultural applications set their mind onto a natural environment, or maybe they were influenced by the way it was explained and framed. We don't know for sure, but we do know that this representation resonated really well with them and it stuck with them as well. Because after the session, we sent out a few questions, like a week after the session, to ask them like what they retained from the session, if they could still explain it in their own words. And this was actually one of the things that really stuck. So they also perceived it as a solution that fits well with an organic approach where they use a lot of natural enemies to eradicate pests. And it does seem like a promising tool for better agricultural practices. But it became clear that the people do not see this as a one-on-one solution as we maybe tend to see it, because they actually came up with another scenario. And so they thought like maybe we should just reduce the need for these solutions on the whole and maybe eliminate the intensive animal production and like do it on a more extensive view, like give animals more space to reduce the spread of the diseases or give them better nutrition and welfare to improve their natural immunity, and combine this with technological solutions for early detections. So especially there was one vegetarian in the session. For her, this was like quite obvious, like are bacteriophages not solving the root problem? So even though everyone concluded that this is a good step towards alleviating some of the shortcomings in the current agricultural system, we do need a more holistic approach in the long run. So the conclusion here is like proceed with care. So even though a few of the participants, like the very progressive one, and were very enthusiastic about this, probably because they look so cool as well. So they were like, what are we waiting for? But most participants were much more prudent and had their reservations related to the broad and massive application, and especially this thing about the mutations. So we concluded that maybe the best approach is a gradual and a partial shift combined with this downsizing and other alternative approaches, because we do not just want to replace one flawed solution with another one. And also that maybe probably more research is needed to learn about the possible impact and side effects of this mass application. Which brings me to my next point, which is that additional research is needed to get a better view of these risks of this mass scale application, especially when you are adding actively releasing like live organisms into the environment in a very much larger scale than normal. And it will increase trust when you get a better view on these possible side and long-term effects. Another question that the participants asked is why that the technologies have not been broadly implemented by now. And there are many reasons, probably. Also, like the previous presentation showed, there are a lot of regulatory hurdles, and there is also a very large recent research investments to be done. And because these are living organisms and you usually found them in nature, they cannot be patented either. So it might sometimes be very hard to compete with the current solutions that are out there and which are cheap and when farmers know how to apply them. So this stresses also the need for investments in public research, because if there's enough information, basic information available, it's easier for smaller companies to turn them into commercial products, because the research phase, the research investment is so fast that it's very hard for small companies to invest in these things. So this is also a good way to prevent monopolizations from large companies who do have enough resources to do all this research. And it brings me also to my next point, is that because people see it as quite a valid and good alternative to current practices, they would really not like to see it hold back because of patents. So people also link patents with monopolies, and they are really afraid of the idea of biotech companies, like the large companies with all the money, having all the power and control over farmers and the food chain. But they do rely on the governments and authorities to install regulations to avoid this monopolization fraud and protect the citizens as well. So this is quite a major issue for consumers. In this case, it was a little less abundant, but it came up in every single session with the consumers as well as with the farmers. So this patenting and fear of monopolization is quite a big issue. And last but not least is that the participants very, very much appreciated our clear and transparent communication. So it was quite a difficult topic, but they were very keen to learn more and very enthusiastic about taking part in this discussion. So they were also quite excited about the information that we provided, and they saw us as a neutral and reliable source because it empowers them to decide for themselves whether these benefits outweigh the risks and how to form their opinions. So this was confirmed by a very small Mentimeter survey that we did at the end of the session. So generally, they understood quite well what was the concept of these bacteriophages, how they work, and what are the risks and the benefits of this technology are. And it also improved the views, slightly improved the views of those who were a bit more skeptical about it. So, and now we move on to the farmers session, and I will give the floor to, or the screen to Heleen, who will talk about them.
Thank you, Charlotte. So I will move on a bit faster. I'm Heleen Imschoot, so Innovation Manager at Flanders FOOD. I have a background as a veterinarian, so this concept I really liked, and I was very happy to be able to participate in the co-creation session with the farmers. So how we set this co-creation session up? Well, we carefully considered the communication. We communicated as neutral as possible with neutral wordings. We really claimed that we wanted to know their honest opinion so they could tell everything they wanted. So it was also important. We created a relaxed environment and atmosphere with coffee, cake, and free tickets to Congress Agri Flanders. So we wanted to create an unbiased open dialogue. So there was space for discussion, and the farmers felt heard and that they could talk about everything. Okay. So the framing of the workshop, we informed them that the session was done in the framework of an European project, and our intention was drafting a method to better include the voices of the public when a legislative framework needs to be set up for new technologies. So here the biotech. We stressed that it was important. They were here because we needed their real input on this matter. So as a warming up exercise, we did two timeline exercises, one with a present and a past scenario. In each timeline, they could mention relevant trends, scientific breakthroughs, innovations, and legislations of the past, future 50 years. Our intention with the future timeline mostly was to get an idea of their future perspective of their profession, their worries and advancements they hope to for or worry about. Okay. Charlotte.
Did it not change?
Okay, yes. Then after the timeline exercise, we did an association exercise. We wanted to grasp the general perception of the farmers on you see biostimulants and biofertilizer, but that needs to be phage therapy, of course. So we provided them with some information. It's a bit different than with the consumers. We provided them with some background about the phage therapy, but we didn't get in depth like we had the presentations now from Damien Thiry and Phageguard. And then last but not least, we did a very important exercise where we talked about the risks and the benefits of this phage therapy. So first we identified the perceived risks and benefits. So to understand what their initial thoughts were on phage therapy, then we presented them evidence-based risk benefits. And so we could have a discussion on that, how these risk benefits based on research were compared to what they identified as risks and benefits. Okay. So their responses. If we look first to get a bit warmed up, how they saw their future. So in the future timeline, a lot of things were discussed. AI, robots, and drones had a big part in their future. Mental health of the farmer, they are under big pressure. They are concerned about. They are also concerned that they will be increasingly restricted by legislation. So it's really hard to be a farmer in practice if there's so many legislative restriction. They are a bit afraid that many companies will stop, so the population of the farmers will age, and they are also quite sure that only big companies will remain because a lot of fusions will happen. In red, you see here some things they said we already can connect to our topic of phage therapy. So here they weren't aware yet, but they already told, so even more feed-related research in function of antibiotic reduction. They were also concerned there will be growing disease pressure in between the animals, and they were quite sure there would be rapid development of new antibiotics. And last but not least, they were also sure that the focus will be on better living conditions of animals in animal husbandry. Then, if we look at the trust barriers they had, so the risks for applying phage therapy, first they talked about upscaling issues. So they find upscaling phage therapy quite unrealistic. We have to be aware they are not informed as well as we are now, thanks to Thiry and Phageguard. So their feeling is it's unrealistic. because a lot of research has been done. They heard about it. They know it exists, phage therapy, since 1914. But for them, it's not clear how the current state of affairs is. They are aware that the disease treatment is very specific, so it's hard to apply broadly. If you compare what they are used to now, so like vaccinations and antibiotics, so it gives a lot of uncertainty, because if they stop doing preventive treatment with vaccination or can apply broad spectrum antibiotics, how will this turn out for them? It's a bit of a risk, and they also think, who will pay for all this changes? Also, who will keep paying for all the extra knowledge we need? So they are also afraid of the safety protocols during the production of the phages, and also about which research has been done on safety and which not. They like to be informed on that. So they really would like to have the entire framework around phages to be first properly in place. Yes. Then another risk, fear of uncontrolled mutations in nature. Charlotte already mentioned this, because you can say that farmers and consumers agree on a lot of topics, and so there's always overlap. So the uncontrolled mutations here, they also mentioned bacteriophages can become resistant to antibiotics. We explained to them that it's true, but the phages can mutate too, so there is a way around, or you combine different strains of the phages to attack the bacteria. So then they were already more into it. They weren't too negative about this. There were options. They just lacked some knowledge about all the options of the phages and all the research that has been done yet. So they were also afraid that mutation would happen to a variant who attacks animals and humans, because they were aware so that the phage recognizes this the cell, and so they compared it sometimes to corona infestations. So what we conclude about this is that misconceptions and fear about therapy with living organisms, we saw it already also in other master classes. This must be addressed through clear communication and targeted education. Yes. So I already mentioned farmers are really seeing the benefits. However, will they harvest them as well? What they see, the positive side, is, of course, this phage therapy will contribute to animal health and a more sustainable livestock farming, reduced use of antibiotics, so less resistance to antibiotics, reduced veterinary costs, because they were aware you could apply it yourself, so the farmer himself, on the animals. So they didn't need a vet this often to vaccinate or to give the antibiotics, so it would reduce their costs. The other part, they were not so happy about, because all the research that has to be done, it's a lot of cost, and we need to produce phage libraries, and that will be a huge cost. But afterwards, they know that the costs are really low of phage therapy if we have those libraries. Also very important, they liked that normally you would have shorter waiting times for the food chain if you use phage therapy instead of antibiotics, because they have quite a long waiting time sometimes. And if necessary, antibiotics would be heavily restricted in the future, then we have a new alternative. So we can say farmers see the benefits of the phage therapy, But they also see still the drawbacks, and they want more education on it, especially on safety. And then they see it as a possible backup or complementary solution to like antibiotics. Okay. So what I also can say, Charlotte mentioned it too, it gives a positive vibe if you say that present the phage therapy in the same sentence as less need for antibiotics. That is really good. You also can say it's a natural treatment. So things like that make a difference indeed also for the farmers. Yes. Then, last but not least, on a European level, the notes to take from the farmers to the European Commission. So, uncertainty of safety and safety procedures, strict standards and regulations should be established to prevent this. Difficult to patent. We had a whole discussion about it. So they were doubting, yeah, well, they are living organisms. Maybe they can be patented, but maybe they can patent the libraries of the phages. So the fear of monopolization is a big issue also for the farmers. Then strict legislation is needed to prevent misuse. So they were a bit afraid the phages would be used as a biological weapon. Last but not least, same as for other cases we saw, farmers expect similar prices to the products they use now, preferably cheaper, especially if they are less reliable, because they have to take a little risk to start with new innovative techniques. And so they want to make it possible, but they can't pay a higher price for it. Okay. I thought we had a question.
Yes, so there's one question. If you consider that there are 10 to the 31 bacteriophages on Earth, how would you define mass scale application in the industrial setting? I'm not sure at who this question is aimed. It was put into the chat when Charlotte was speaking. So I think, Charlotte, you also talked about mass scale application. Could you answer this, or is there another speaker who can provide an answer?
I think I can answer that. Because mass scale application is like mass scale application of one single or a few, very few bacteriophages of the same kind. So there are 10 to the 31 bacteriophages on Earth, but there are all kinds of different bacteriophages. But once you go and apply a very single specific one, it will have an effect on how they are balanced with each other.
Yeah, can I add? Do you hear me? Or yeah.
Yeah.
I like the little bit of the discussion also. And for example, in this respect, you could make the comparison to probiotics. Probiotics that are also widely produced in, well, all sorts of dairy, drinks, etc. And then you also basically implement a lot of organisms in the world. But that would possibly not be expected to be resonating negative, right? But it is always good to consider the context, I guess.
Yeah, probably not, because indeed it was taken together, this case, with the biostimulants, which is more like probiotics actually. So, and they did not see the same harmful or same risks as with the bacteriophages. So this,
because if you would say, hey, we gonna produce a lot of chemicals in the world, people would also be a bit worried, right?
Yeah, sure.
Yeah.
This is why they also saw it like still a valid option, because the alternative is not that great either. So
yeah, that's interesting. Thank you.
Thank you. Are there any other questions?
Ask one question.
Yes.
To Jeroen and also to Damien. But I heard Jeroen say, yeah, well, we have educational programs. Do you also do educational programs for the farmers?
Thank you, Heleen. No, we are not so much oriented towards
No, I know.
the primary industry, but we team up quite a bit with educational organizations, mainly training the QA managers of the future, so to say. So that is still pretty technical, but we also often participate in open days or new science type of experiments to expose the public, so to say, a bit more to bacteriophages. But in general, the activity is very much technical in education. Yeah.
Understand, but if it would be possible to apply them in veterinary science, you probably would also educate them because they are. I just wanted to stress that they really want knowledge. I think if they saw this presentation, the risks would drop down very fast. We presented. So Damien, I don't know if you know about support for the farmers, about knowledge about those.
No, but it's always the same case. If you don't have the product on the market,
I know.
you will not have no information will be given to the farmers or the vets, etc. It's difficult to advertise something that do not exist in the field. So that's the same case for the vets. If we want to make some continuing education for vets, and we want to implement a phage lecture, it's difficult because as you don't have phage in the market, you don't have any. Okay, there is some interest, but not a lot. So it's difficult to make advertising of something that do not exist. But when it's really interesting what you presented, the two last studies, because I think that I understand the fact that having experts or people who are maybe too strict in their opinion on the topic could be an issue in this kind of discussion, but sometimes it could also help to answer some questions that are really interesting, because what you presented now is really interesting, and the reflections are really good for most of them. The questions that they raised are really good, and we can discuss them. What I would like to comment here is that, to my opinion, we have to consider phages as a tool. This is a tool in a toolbox, and this toolbox contains many tools that will help us to fight against antimicrobial resistance. We will not fight against antimicrobial resistance with only one tool. We will fight against with several tools. And when you talk, for example, with vaccination, you cannot imagine to replace vaccination with phage.
No.
And this is not the aim. Of course, if there is a good vaccination against the disease, the farmers have to vaccinate with this vaccine for sure. But we can imagine phage for certain specific disease for which we do not have any solution or not good solution. The example that I gave with post-weaning diarrhea, because I can talk about a lot of disease. We also work in fish, etc., or companion animals. But if we talk about, for example, post-weaning diarrhea, in the past, the vets used a lot colistin, and colistin is now a last resort antibiotics for human medicine. So, and there are a lot of resistance against this antimicrobial. So, farmers and vets cannot use anymore colistins or with really specific rules. They also use to prevent this disease zinc oxide. Okay, zinc oxide has a huge impact in the environment. Okay, so they banned this use of zinc oxide in the feeds of the piglets. So we need some solution, and there is some vaccine, but not good vaccine for all the type of ETEC. So for this kind of disease, I think phage can help, not solve the issue, but can help. So I think that's why I start my presentation with, I do not like, I don't know, I don't remember if I started or if I finished the presentation with this, but I do not like the terms alternative. For me, it's not an alternative, it's a complement tool.
I think it was quite all right how they perceived this, but I just told this more in the frame of we are here to create public acceptance for biotech. And even we don't apply the phage therapy yet, it would help in the acceptance because they are non-aware, and you see that this is quite an obstacle. And the farmer really feels like he's kept a bit dumb. They're really interested in how everything functions. And they have a lot of questions. Also, farmers are with their feet in the ground. They are sometimes a bit fearful, but they are also quite realistic. And qua management, to manage in the farm, like how do you apply? Do you put the diseased animals all together in one room to apply the phages, things like that? That is of their interest.
Yes, and this is really a good question, and this question are really difficult to answer because, of course, when you talk about mass treatment, it's difficult to imagine for the moment a mass treatment with a phage into the feed, for example, because it will cost a lot, for sure, to give this kind of treatment. So, but if you think about, for example, bovine mastitis, if we can have a good cocktail of phage for bovine mastitis, it can be it can help a lot, because we know that vaccines are not always okay, etc. So then you have a really localized treatment for some animals. So you see, we can imagine like that. But I agree with you. The communication is really important, but I do not say that I'm not for the communication, but it's difficult to convince people who are, so that's why. And another thing is about patent. I would like just to comment. Of course, it's difficult to have patent with phages, but people have to be aware also that without the companies, we are nothing. We cannot have a product on the market without companies, or it's really difficult because we need companies that invest in this option. And if we don't have the companies with us, it will be more and more difficult to get this kind of product in the field. And that's also why there is no phage currently in the field. That's because companies do not see their interest to develop this kind of treatment because it costs really a lot to develop it. So it's true that patent can have a kind of monopolization, but we have to see the balance also.
Okay, thank you so much for this. If they view this afterwards, it will be very much appreciated, I'm sure. Thank you.
Thank you for all the questions, for the discussions, and also for the lovely presentations. Yeah, I was happy that all of you could join us today, and hopefully we will see you at the next masterclass.
Thank you, and highly appreciated your activity. So well done.
Thank you.
Thank you.
Bye.
Okay,
Bye.
bye-bye.